Welcome to the Health Tech Invest podcast powered by Nutera Ventures, your guide to healthcare investing. Join us as we explore interviews with pioneering entrepreneurs, investors, and innovative leaders, helping you spark innovation in the world of venture capital investing. Awesome. Thank you so much for joining us on Health Tech Invest with Tom Clues. I've got a very exciting guest for everyone today. The name is Dr. Chris Murphy. He is an ex-professor at UC Davis. He is a distinguished professor emeritus for the Department of Ophthalmology at the Schools of Medicine and Veterinary Medicine at University of California, Davis. He is also the CEO and founder of Ocular Services on Demand, a merit company, and he is a principal and co-founder of a number of innovative companies, up to and including one of our portfolio companies, Imbed Biosciences. Chris, pleasure to have you on the podcast today. How are you doing?
I'm doing great, Tom. Thanks for bringing me on. I'm intrigued by seeing what questions you ask. I'm intrigued.
Awesome.
And by the way, this is the first podcast I've ever engaged in.
Awesome. This is great. You're a podcast. This is one of the few things that you're doing for the first time. So I actually we'll engage in the journey together. So thanks for being on the podcast. So we're really excited to have you, Chris. I think I didn't give your background justice here, so I thought best way to kind of kick this off is for you to give our listeners out there a brief overview about your background and tell us a little bit about yourself.
Okay, so a brief journey through time and mention things that I think are relevant to who I am. I am a New Yorker. I was born and raised in New York. I'm the middle of 7 children. And that's important because when you have a large family, you are raised by your siblings as much as by your parents, and affects much of how you traverse life. I went to 3 undergraduate universities, and I ended up at Cornell, where I stayed for 10 years, and I got my BS, my DVM, and my PhD at Cornell. I then met my wife, my lovely wife, Joanne Paul Murphy, before vet school and before my PhD, the end of my undergraduate degree, and we got married, and she's also a veterinarian and distinguished in her own right. In her field, I'm best known as her husband. And then we went from New York to California, where I did my residency training in comparative ophthalmology. I did a 3-year residency. And at the end of that, I have a little bit of a unique attribute in that I then did a Cornell fellowship. As far as I know, I am the only veterinary ophthalmologist to do a formal fellowship at a medical school where I saw human patients for 2 years. And at that point, I also did research and got involved in wound healing from the cellular molecular basis under the tutelage of Ted Reed and Mark Manus. Mark Manus, who was then an assistant professor, later became the chair of ophthalmology and is a worldwide renowned corneal specialist who just recently has stepped down as being the active chair of ophthalmology at UC Davis. So then we were there, and after I finished my fellowship, I was then recruited as an assistant professor at the medical school, and I was cross-appointed at the vet school, and I was in private practice 2 days a week because I wanted to keep my skill set up and microsurgical training. Because of my corneal fellowship, I got advanced training in microsurgery. And after 2 years, Joanne and I migrated from Davis. We went to the University of Wisconsin in Madison, and that was in 1991. We stayed at Madison for 18 years, and lots of things happened in Madison. I rose up through the ranks to achieve the rank of full professor. I started a very large laboratory. It started off with myself and one person. When I left Wisconsin in 2009, I was running a well-funded, federally funded laboratory of 25 people, focused predominantly on wound healing and biomechanics of wound healing. And in that period at Wisconsin, I then started finding that I really enjoyed business. And I founded my first company was in 1993, Ocular Services on Demand, which I am still the CEO of. That started with three of us. That was co-founded with myself, Dan Albert, and Dick DeBilzik. Started with three of us because we realized we had a world-class ophthalmology section at both the med school and the vet school. The three of us were doing industry consulting when we realized there was a niche that had not been occupied of integrating academic expertise to benefit drug development in the eye. So we started off with three, and today we have about 60 members. We have 10 full-time employees, and we grew organically in that whenever we had a problem that we did not have the expertise to address, I reached out, interviewed, got opinions, sought out, and integrated the next member. It's kind of a running joke. I took them to lunch and then they drank the Kool-Aid. Because to take an academic and bring them into an industry project, they had to agree that the industry project was a priority, that it was not priority 25C behind their papers and their students and their postdocs, that they had to make it a priority, and that they never had to pick up a project. If they were too busy, it didn't fit in, they didn't have to pick it up. But if they picked it up, they could never fail. They could never walk away. And with that understanding, we grew the company, again, one person at a time, now over 60. And we had a seminal event in Wisconsin where we were working then with Covance, which is a CRO, international, multi-billion-dollar company, is in Madison, Wisconsin, headquartered. And we developed a relationship there. And I can talk about this because we have published on this. And part of the thing when you're working with drug companies is confidentiality. You can't disclose ever who you're working with or what you're doing. But I can talk about this because we published on it. So we were part of the scientific team that worked with Genentech and Chuck O'Neill, and his name will come up again later as we talk about things. Chuck was the lead preclinical toxicologist at Genentech responsible for developing a drug called Lucentis. And we were the scientific arm supporting him at Covance. And that was a challenging project with lots of issues that had to be addressed. And OSODs went from 3 of us to about 15 of us by the end of that project. Because we had to integrate pathologists, we had to integrate clinical scientists, we had to integrate immunologists, and we had to integrate advanced imaging, advanced electrodiagnostics, all of that in there. So while I was in Madison, I was a co-founder of Ocular Services on Demand. I was a co-founder of Platypus Technologies. Platypus was founded on innovations on liquid crystal reporting systems for cell and molecular and aerosolized interactions with nanostructured surfaces using liquid crystal as a reporting medium. And it was in that context that I became involved with biomedical engineering, chemical engineering, and indeed I became cross-appointed in biomedical engineering as a professor. And I helped train many chemical engineering and biomedical engineering PhD students and postdocs over the 18 years I was in Wisconsin. And there were several other companies that we'll come to that happened, including Imbed, which happened right when I left Wisconsin, which was in 2009. Imbed was founded. Ankit Agarwal I was on his mentoring team along with Nick Abbott. Nick Abbott is a co-founder of Imbed. Chuck Saprinsky is a co-founder of Imbed, all at Wisconsin. And so in 2009, we came back to UC Davis, where I was appointed as a professor in comparative ophthalmology with a primary appointment at the medical school and cross appointment at the veterinary school. And I was responsible for training residents in microsurgery at both the medical school and the veterinary school, clinical comparative ophthalmology at the veterinary school. And I established a large lab with Paul Russell, who had been at the National Eye Institute for 30 years before coming to work with me in Wisconsin. And when we landed in Davis, we came in with one postdoc, and in 12 months, we had a lab of 30 people.
Wow.
That we had hired everybody, and we had multiple NIH grants at that point. And then we got many more NIH grants and NSF grants as we were going through. So that's a brief history of time.
Awesome. Awesome. So I have so many questions. I think I'll start with this this dual appointment that you had at UC Davis with both the School of Medicine and the School of Veterinary Medicine. I question there is what drew you to that? What drew you to ophthalmology, and how did your career evolve across all these diverse fields?
So there's several questions to unpack there.
Yeah.
So what drew me to ophthalmology is sure happenstance. We like to think that we've got some plan, and we present our life story as if it's linear. It's chaos. It's chaotic. And I knew I wanted to be a veterinarian from the time I was 8 years of age. I went to Cornell to become a bovine practitioner. I had worked on farms, both dairy farms, and I had worked in equine practices as a vet tech. Not a certified vet tech, but interested college student interested in veterinary medicine. I'd spent a year doing equine medicine as a technician. And so I met my wife, who's pivotal in my choices here. Because I met my wife, we both got accepted in the veterinary school at the same time for the same class. And Joanne, honestly, was a better student than I was. And I was sure she was going to get in. Wasn't so sure I was going to get in. And I was positive I would become a veterinarian by hook or by crook, because I am, if nothing else, I am tenacious. When I set a target, I have a tendency to achieve it. But I'm also a realist, and I'm like, Joanne's getting in, I may or may not. So I applied to graduate school to cover my options. It's good to, as you know, in business, it's good to have optionality. In life, it's good to have optionality. And so I got accepted into a master's program to study comparative medicine, comparative anatomy at Cornell. And it was not at the vet school. It was in the field of zoology. I was accepted as a master's student. Well, I had a very curious and very defocused primary mentor who would come to me and give me a project, and it would be unworkable. It would be, I'd go off and I'd spend 2 weeks looking at it, and I'd be like, this is not possible to do this project that you want me to do. And I'd go and meet with him, and I'd give him what I found and why the question he wants to answer is not going to be able to be answered in the 2-year time frame of a master's. And we didn't have the resources, the instrumentation. And then he'd say, I got another question. About the 5th question that he said I could spend time in was the anatomy of the raptor eye. Raptors are birds of prey. And that really resonated with me because I had worked in zoos as an undergraduate at Cornell. Joanne and I both worked at zoos. And Joanne and I were both employees of the Peregrine Fund at Cornell, which was then establishing the peregrine falcon population east of the Rockies with a very famous ornithologist named Tom Cade. And so we were working with master falconers. I fell in love with birds of prey. And so I thought this would be perfect 2-year project. I would be able to get a master's, and I'd get to work with birds of prey, and I already had connections there, made a lot of sense. When I started doing that, what I found is I love doing research. And I've always been curious. And when I figured out that I could maybe make a living by being curious and answering questions I was interested in, game over. I knew what I wanted to do. And I turned my master's into a PhD. And so I had deferred my acceptance into in the midst of all this, I got accepted into vet school at the same class that you did. And so to defer, and then we decided it would not be good for our new budding marriage to be in the same class, because it's vet school, as Tom, is intense, and it's highly competitive, and we didn't think it would be the best thing for our marriage. And maybe one of the most adult things I've ever done was to defer acceptance into the vet school, but they wouldn't let me defer. But when I wrote this erudite letter about why it was such an honor to be able to study with Dr. Howard Evans, one of the world's foremost comparative morphologists, especially well-known in veterinary medicine. And they said, no, you got to come. And he went down to the dean's office and to the board that refused my request and said he doesn't want to be in the same class with his wife. And they were like, what? What wife? Because Joanne's maiden name was Joanne Paul. They didn't connect that Joanne Paul and Chris Murphy were married. So they immediately granted my deferral because they were like, I don't want to be in class with my wife either. So because of that, I got to do a master's into a PhD and change my career trajectory. And when I did it at Cornell, it was not a conjoined program. I was in a PhD program in zoology in the biological sciences, and I was in a veterinary program in the College of Agriculture. And they were not interjoined. So I had to literally resign from my PhD every year to go to vet school. And I had to step out of vet school to go and do my PhD for the year that I did before vet school. Every summer, I had to resign from one school and enter another. And I did that throughout vet school. Every Christmas break, summer break, Easter break, I did my PhD work. And then I did my PhD the first year before vet school, all throughout vet school, doing this as I described it, and 1 year after vet school. And then, so I got my DVM, my PhD out of 2 different programs at Cornell in a 6-year period. And then,
Impressive.
When I found out that I could make a living doing research, at the end of my PhD, I was offered a number of academic jobs in comparative because I was trained at that time, was the premier comparative morphology program in the world in veterinary medicine. And I decided I wanted to be a veterinarian for a reason, and that was to see patients. So then I did my clinical residency, and that led to doing the Cornell Fellowship, which then led to a combined program. And I was cross-appointed at the med school in both Wisconsin so the med school, biomedical engineering, and the vet school at both Wisconsin and at Davis. And that's
interesting.
And why the med school? Because I had done a Cornell Fellowship. I had seen human patients for 2 years. And the beauty of ophthalmology is that it is one of the most conservatively structured organs in the body. Every vertebrate on the planet has an archetypical design that is invariant.
Interesting.
50,000 vertebrates, 50,000 plus vertebrates on this planet have an eye that has an outer coat, a middle coat, and an interneural coat, and the internal optical media, the aqueous humor, the lens, and the vitreous body. So humans are just another species.
Interesting. So what I guess what makes them Because I've heard birds have very complex eye structures. They have the ability to accommodate kind of nearsightedness, farsightedness. Again, and I'm not going into sort of the details of this, but can you talk a little bit about the differences between different
Yeah.
eye accommodations?
Sure. About accommodation or about structure of eyes?
The differences.
Yeah.
I mean,
Okay.
it sounds to me structurally, they're all pretty similar, but
Well,
then it's the application that is where it changes. And I don't know if that's the right wording, but
no, so, I give a whole lecture on Joe vertebrate eye.
okay.
Joe
Okay.
vertebrate eye has am I allowed to pull pictures up here? No.
You can. Yeah,
No, I'm not.
absolutely.
So
Yep.
Joe
Yeah.
vertebrate eye is an outer fibrous coat that's composed of the cornea and the sclera.
Okay.
The middle vascular coat, collectively called the uvea, that is comprised of the iris in the front of the eye, the ciliary body surrounding the lens, and the choroid that underlies the retina. And the interneural coat, which is the retina of the eye. And the internal optical media, the aqueous humor, which is between the iris and lens and the back of the cornea. The lens itself, which is behind the iris and suspended by the ciliary body of the uveal middle vascular coat. And the vitreous body, which is the biggest single structure in the eye, which is 98% water. It's got a lot of hyaluronic acid in it. It's got a lot of glycosaminoglycans, such as hyaluronic acid. And that is lying between the retina itself and the back of the lens. It's a big jelly structure. It's kind of squiggly. So every vertebrate has that structure. Now, if you want to make a fish, well, nobody can take away any of the Joe vertebrate eye structures. How we make different species is we add things or we make specializations. So if I want a fish, I'm going to add some bone in the front of the sclera, integrate it into the sclera. I'm going to have a very spherical lens. Why a spherical lens in a fish? Because fish live in water. What happens when you stick your eye in water? You all of a sudden can't see so well until you put an air-filled mask over your eyes. Why is that? Because your cornea has the same refractive index as water. So you lose the optical power of your cornea when you immerse your eye in water. Well, fish live in water. Don't need no stinking curvature to that cornea because there's no power to it. And it actually adds drag energetically to have a curved surface. So they have a flat cornea. So now they have no power in their cornea, but they still have to image the world on the retina. How do you make up for the loss of the cornea? You make a spherical lens. They don't have any blood vessels in their retina. So how does the retina get its nourishment if there's no blood vessels? Well, they get it from the choroid. Everybody gets it from the choroid, but they also have accessory nutritional structures such as the falciparum process and a thing called the choroidal gland, which is associated with the choroid. So that's Joe fish. Okay. And there's lots of nuances to the structure. If you want to make a bird, you put bone in the sclera that determines the shape of the eye. You put striated muscle in the iris, striated muscle in the ciliary body. Striated muscle is the kind of muscle you have in your biceps. In a human and in mammals have smooth muscle. So smooth muscle versus striated muscle. Smooth muscle contracts slowly. Striated muscle, like a snap.
Interesting.
The pupil can move very, very quickly. The striated muscles in the ciliary body can change the shape of the lens very quickly, which makes it very fast for changing focus. Think of a peregrine falcon going at 200 miles an hour, chasing a pigeon at 60 miles an hour. And how do you focus on that? Because if you err on your depth perception, you kill yourself hitting the pigeon.
Yeah.
You know just when to pull up. So, and birds don't have any blood vessels in the retina. They have a specialized structure called the pecten oculi. That is a big vascularized structure that sits into the vitreous and provides nourishment to the retina itself. So those are just a couple of examples as to how you build these different eyes. But they all have a basic structure that you then add to. You specialize upon, and makes it so that every ophthalmologist can look at any species and know what they're looking at.
Interesting. So I have a fun question here. So true or false? Some fish can see better than humans underwater and in the dark. True or false?
True.
Okay. So tell us about, so tell us a little more about that. What's the deal with aquatic vision? How do fish see underwater?
The example you brought up is not about being aquatic. It's about living in the dark. There are many species that live in the dark. So when you talk about in the dark, you can go to fish that that are active nocturnally, or you can go to fish that live at depth because there is no light that penetrates the oceans. So you have specializations similar to an owl, which has a tubular shaped eye, which allows for tubular shapes happen when you want to have a really big eye, but you can't afford to carry all that weight. So you carve out the edges and you tubular shape. And owls have a tubular shape. Other nocturnal birds, such as caprimulgiforms, which are like nightjars and whippoorwills, have a tubular shape. And if you go to deep sea fish, they have a tubular shape. That allows for a big pupil, better for collecting light. And generally, if you're operating under extreme, we would use the term scotopic conditions, dim conditions, you generally have a retina that is specialized by having a lot of rods and not many cones. And then in terrestrial vertebrates, you would have adaptation in the back of the eye associated with the vascular coat of the eye in most mammals called the tapetum lucidum. So if you drive down the road at night and you see a deer with headlights and its eyes are green, or you look at your cat or you look at your dog and the eyes light up, that's reflection of light coming off from the back of the eye. What does that do? Because that mirror-like structure is located behind the sensitive retina, the light has to pass through the retina, it hits the mirror of the tapetum, bounces back through, and it gives the retina two chances to collect the same quantum of light, increasing. It means that your dog sees better than you do at night. The horse sees better than you at night. And most herbivores will have, like goats, deer, cows, have tapetum lucidum. This is a board question that we ask veterinarians. What domestic animal doesn't have a tapetum? The pig have a tapetum. So, yeah. So those are adaptations, and it's part of the fascination on how you take this structure that seems pretty homogeneous in terms of its basic architecture, and you add nuances to it. It makes it interesting as a scientist. And as an ophthalmologist, we get to see beautiful eyes all day, every day, not monkey after monkey after monkey, which is what you are doing if you're a physician ophthalmologist. It's a primate after primate. Now, primates, great. I love primates, but I prefer being a comparative ophthalmologist as a veterinarian.
That's awesome. So look, I've seen these kind of memes online where, what would humans see if you had the eyes of a hawk or a fly, right? You've got super zoom, 360-degree vision, ultraviolet colors, right? How accurate are those memes? I mean, can you talk a little bit about that?
You mean are you talking about the programs or the memes? The images?
Yeah. So I see these images. I'm like, if I had the eyes of a bee, I would see 360 degrees around me, and I would have ultraviolet vision. I mean, are those like accurate depictions of what's going on? Are they scientifically backed?
Okay, that's a deep question.
Yeah.
The answer is yes, and the answer is yes, and the modifier is partially.
Okay.
So we can ask, what can an animal see?
Mm-hmm.
So, and there are very famous experiments done at the turn of the century by Carl von Frisch in
Mm-hmm.
Germany. And he really laid the groundwork for understanding insect vision. And
Mm-hmm.
he studied color vision and visual capabilities in bees.
Mm-hmm.
And he was able to document what is the spectrum that the bee can see.
Mm-hmm.
And yes, and there are many animals that can see into the ultraviolet. And there are birds that can see into the ultraviolet. And there are many insects that can see into the ultraviolet range. But what is the experience of perceiving in these animals
Yeah.
is
Yeah.
where we say, yeah, yes, it's true, but are we really capturing how they experience it? We're taking our best guess because we know, for example, you can take the woodcock, this little bird, beautiful little bird. Its eyes are located exactly lateral in the head. They have a binocular field that's very small in front of them
Mm-hmm.
and behind them. They see 360 degrees with a very small overlap of binocularity. Why is that? What's up with that? Well,
Mm-hmm.
they're a prey species.
Mm-hmm.
And when you think of evolution, you want to think of ways not to be taken off of the gene pool, because the only way you continue to populate your genes into nature is by reproducing. And of course, if you're eaten, you can't reproduce anymore. So that's a big pressure. So when you look at all of these different species, what you have to think of is that every species has answered a series of challenging environmental questions that is the best answer they could come up with. And if they are around and in significant numbers, they have
Uh-huh.
answered the challenge correctly.
Uh-huh.
So, when I was being brought up, I was lied to by my nuns that taught me in parochial school that animals were colorblind. They're not colorblind. Are there some animals that are colorblind? Yes, there are.
Mm.
They have only monochromatic vision, only in black and white. It's rare to have monochromatic.
Really?
Most animals have less color vision than humans. We're very anthropocentric. If people say, "How well does name a species see?" We all want to know relative to me, right?
Yeah.
It's a little bit like when you look in your family portrait albums. Everybody's looking for their own pictures. They're
Mm-hmm.
not looking at
Mm-hmm.
everybody at cousin Joe and second cousin Sarah. Everybody wants to see the pictures that they're in. And when
Mm-hmm.
we ask questions about how well anything is happening in nature, it's almost a fundamental to say we want to know how well is that animal relative to me. So that's a hard question to answer because many people think that vision actually has one attribute. Vision has many, 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 many attributes. You already brought up some of them. One is how big of a visual field can you see? Owls are like us. We're facing straight forward. We've got great binocular fields. Why is that?
Mm-hmm.
Because when you have great binocularity, you have great depth perception. And we are opposable thumb primates. And we make
Mm.
moving things around with our little fingers. Well, if you're a horse with hooves, you're not doing
Mm-hmm.
it like that.
That's
You know,
true. Yeah.
what you want to do is make sure you're not eating. You don't have to chase any grass down. It's
Mm-hmm.
on at your feet. And really your evolutionary pressure is to not get eaten, right? So every animal will have their answer to the evolutionary pressures, the ecological pressures, the ecological niche that they occupy. Their answer is perfect for them.
Yeah. Yeah.
You know?
So you bring up a really interesting point earlier about, it's one thing to have sort of the anatomical piece, but then the other is the perception of that vision, right? I know it might be a bit of a tricky question, but where is science today in actually being able to measure the perception of how one animal sees something versus another?
We're a lot further along, but I'll
Hmm.
tell you, remember I brought up the number 50,000 species
Hmm.
of vertebrates. Vertebrates, just vertebrates. I'm not talking invertebrates. There's millions of invertebrates. 50,000 species of vertebrates. There are 30,000 species of fish, 10,000 species of birds, 6,000 something species of mammal. And then you get into amphibians and you get to reptiles. There's a lot of vertebrates on this planet. How many have we really studied? Very few. When people talk about big comparative surveys, what are they talking about? They did a mouse, a lab mouse. A lab mouse is not a normal mouse. That they had no pressure to survive. We bred the
Yeah.
individual capabilities out of them. Same with rats. And some of these studies have been done. If you get a wild mouse, their visual capabilities are different than a lab mouse. So where are we now in understanding? We're way further forward than we had been, but we know more about the vision of the celestial goldfish or the cat, domestic cat, than we do about most of the species on this planet. Why is that? Because cats have been used as a model for neural processing in humans. Because again, we're the most important species. Just ask any other human, right? And we have tools available now that were not available 50 years ago to ask questions like, what is the local visual acuity across the retina of an animal? And we can do electrodiagnostic testing using what's called a multifocal ERG, which basically allows you to do electrical testing at hundreds of points across the retina, not just a full field thing, which is what flash ERGs give you. And we can, how well does each part of that retina see in terms of its ability to resolve 2 points in light of 2 points in space as being separate from each other. So the tools are a lot better.
Wow.
Again, we're going to come back to how does the animal integrate that? And then you get into like wolf spiders and jumping spiders with eight eyes, and the eyes have different structures, different capabilities, different wiring. And even in humans, just to remind you that what we see is what we think we see.
Sure.
Because we have rays of light entering our eye, we see only things that light that passes through our pupil. We don't see all the stuff going on around us. And we take an object, and if we put it 20 feet away, only parallel rays of light can enter your pupil. Because if it was diverging or converging rays of light, will not enter a pupil of an observer 20 feet away. So from 20 feet to infinity, it's all the same because it's parallel rays of light coming into you.
Interesting. So does this mean, so you bring up a really interesting point. So it's a powerful idea. Do you think, so let's talk about what we think we see. Do our brains shape the reality of what our eyes are seeing? Is that what's going on here?
Because where I was going with this is we have our retinas, we have images, we have rays of light that are brought to a focus on our retina. They then are processed. The information's processed. A phototransduction cascade in your rods and your cones generates an electrical signal, goes back towards the vitreous, goes from the photoreceptor out to the outer part of the eye. Then it goes to the inner neurons where it's processed. And it goes to your ganglion cells. And your ganglion cells basically set off an action potential, a little pulse of electricity. It goes down this axon. All the axons are gathered together. That's your optic nerve. That has to go up. They cross over partially at an area of your brain called the chiasm, and it crosses over from your right to your left. And some of it stays on the right side, some of it goes to the left side. There's species difference in how that happens. It then goes to multiple centers in your brain to end up at your visual cortex, which in mammals is in the back of the eye, back of the head.
Mm-hmm.
Well, all that processing, what we're seeing is the result of millions of little action potentials being processed through your brain, integrated at your visual cortex. And that's why we believe what we see is real. Now, there are things all around us. We have a narrow part of the electromagnetic spectrum that we actually can see. There are animals that can see wider parts of the electromagnetic spectrum, like ultraviolet. Humans don't see it because they don't have the photoreceptors set up, tuned to that wavelength. And then it gets even more interesting when you think about the ability of animals that have senses that are alien to humans, where they are perceiving their environment using receptors that we don't have, whether it's electrical reception. There are notoreid fish, night fish. If you go to an aquarium, look at the night fish or the elephant trunk fish. They generate electrical fields that they can then have receptors and can sense three-dimensional electrical fields, conducting and insulating elements in their environment. You can have the Pizza Lorenzini in sharks are receptors, electrical receptors that they can detect prey in sand, electrical signals. Lateral line system of fish like trout, they can perceive pressure differences in 3 dimensions around them. That's stuff that we have no idea about. So and then you can get to the infrared receptors of rattlesnakes and pit vipers, where they can perceive heated objects in 3 and react to it. They will strike. I mean, this was a thing we did as undergraduates. You put a warm balloon in a blindfolded python in front of them, they will strike at it because that is prey.
Interesting. Yeah, I mean, it's crazy, right? So our brains, our brains are really shaping the reality more so than the eyes are, right? I think the physical limitations of the eyes probably control the type of signal the brain receives.
That's the sensing element is the eye. And the integration is through central processing.
Yeah.
And it's ultimately, it's a series of electrical pulses that set into the brain, process, ends up at the occipital cortex for integration. And our brains lie to us all the time. We see things, we fill in blanks, we expect things, and there are differences that we see. And to give you an example, if you were to cross over your auditory nerve into your brain with your optic nerve, you would hear the lightning and you would see the thunder.
Yeah. That's crazy. That's crazy to think about. Let me ask you a bit of a philosophical question. So, if perception is kind of, and obviously it's filtered through belief or expectation, right? How do we ever know what's really there? Right. And I've had this discussion with friends in the past, right? So I know what red is because I've seen red before, but somebody else might see it a little bit differently. How do we sort of bridge that gap?
They do. I can tell you that my visual capabilities are not exactly the same as yours. How you perceive color hues may be a little bit different than mine. We arrive at consensus. There's a whole field of philosophy. I'm glad you opened it up. The field of phenomenology, very big in Germany, with Heidegger and Husserl. And there was a science to doing what they called the phenomenological reductions. We can't truly know exactly what the other person senses, but we agree that there's a commonality. And scientifically, we can show that what you see as red, I can see as red. You see as blue, I can see as blue. And keep in mind, 8% of all the men on this planet are red-green colorblind. right?
How many? What percent?
About 8%.
Oh, 8%. Okay. I thought you said 80. Wow. Okay. That's
They,
okay. Very cool.
and there are
Yeah.
some interesting studies where there are islands that have
Mm.
very high percentages.
Interesting. Interesting.
And also, interestingly, there are some primates that don't have full color vision. Humans have trichromatic color vision.
Mm-hmm.
Dogs, for example, most mammals
Uh-huh.
they're dichromatic.
Uh-huh.
They're red-green colorblind. There's a group of primates, and this is, if you want to read something, pick out a book and some of the papers by Jay Knights at the University of Washington, UC in Wisconsin, which is where I met him. He's a world authority. He and his wife are world authorities on color vision and in animals and in humans. And he documented that he could introduce a gene into an animal that normally can't see a color and introduce a gene into their retina, and they can see that color. And experimentally was able to document that, which has consequences for people who are red-green colorblind.
What's the name? Sorry, just for the listeners out there. You said JN.
Neitz.
Got it. Very
He's
cool.
also a very nice guy. And his wife, Maureen, is a cell molecular biologist who focuses on color vision.
Interesting. So we're really far, I think, scientifically from being able to say, hey, like we can so we can trust what we see based on these commonalities that we've kind of objectively measured. But when it starts to come, when we start to look at sort of the nuances of that, I think we're a bit far away from being able to objectively measure things, right?
Well, I think for humans, we know a lot. We know a lot about what humans can see. And I could bring you into a clinic and we could figure out, do you have limitations? Do you have the normal amount of vision? And it's really about what's normal. And what's normal is the statistical normal. Like when you say you have 20/20 vision, that means that at 20 feet away from a target, you can distinguish 2 points in space as separate, as well as a statistical average human, taking a sample size. So you might be 20/20, and that'd be what's called your best corrected visual acuity. I'm 20/15. What does that mean? It means that I have better vision for acuity than the average human, because at 20/15, an average human has to be at 15 feet to see what I can see at 20 feet. Now, if you have, and so it's based on comparisons to what is, quote, normal for an average person.
Very cool. That's awesome. No, this is fascinating. I could go on for hours here talking about eyeballs. But the last question I'll ask you on this topic is in all your years of vision science, what species, what animal has the coolest eyes, or most impressive eyes in your mind?
Well, I can tell you it's whatever animal I'm looking at.
Okay, cool.
Every animal has got unique attributes. There are, attributes in like freshwater turtles, red-eared sliders. They have all these crazy color droplets in their cones, all different colors. And what's up with that? Well, they have intraocular sunglasses. They have intraocular color filters of different colors that are tuning the light rays that hit the cone photoreceptors. That's perfect for what they're doing. And then they also have polarizing vision, which is good. Just like we put our Ray-Ban polarizing filters on when we're out because there's glare at the water. Well, think about a turtle. They're living at that interface. So they got polarizers.
Turtles don't need their Maui gyms.
Yeah, exactly.
They've got them built
That's
in.
exactly right.
I love it.
There are animals that, when you look at the lids of, for me, hornbills, the bird hornbills, have these unbelievable lashes. You just can't, and they're not really hair. They're feathers, called biloplumes. Unbelievable looking. You can look at pupil shapes. There are papers from the turn of the century with just the pupil shapes in frogs. You can think of geckos. Geckos have under bright light conditions, they have 4 separate pupils. Well, with 4 separate pupils, that has optical consequences. They get
Mhm.
a split prism focusing device by having four separate pupils. So there's amazing adaptations everywhere you look in nature in terms of the visual system. So again, I'll come back to what's the most fascinating? The animal in front of me. Whatever animal that is, there's a story to tell.
That's awesome. I love it. I love it. No, thank you so much. That's pretty cool. That is super cool. Let's change gears a little bit and talk a little bit about your kind of shift from, I wouldn't say shift, it's just your experience in entrepreneurship. I feel like you've never really transitioned from one to the other. You've done both, right? You've been an academic, but you've also built and scaled companies. Can you tell us about what drew you to entrepreneurship? What is it about entrepreneurship that made you pursue it?
Well, when you do anything and you do it intensely and you do it well, you get to a point where you feel like you've sort of seen 80% of what you're going to see, 90% of what you're going to see. And I sort of got to that point when I was at Wisconsin. I had built a lab. We were successful in research. We had grants. We were answering questions I found interesting. I had given heaps and heaps of lectures. I had trained many residents. I had trained, at this, now that I'm my age, thousands of veterinary students. But at that time, it was, let's see, probably over 1,000 veterinary students at that time. And I started getting this opportunity to start doing consulting as an ophthalmologist for industry. And I found it interesting. And when I saw that there was a niche that we could fill, and I had people around me that I could do something novel that I thought would do some societal good, help patients. My motivation, the hat trick for me to be engaged in something is the following: societal good, great teams, some profit. And
I
if
like it.
all three of those are in place, it's hard for me to walk away.
Mhm.
Now, can I do something that's really incredibly societally good
Mhm.
with an okay team and no money? Yes. But the balance, that hat trick, that's the hat trick for me. The three legs of the stool in which I support how I engage in entrepreneurial activities. Societal good, including help, a big driver for that is helping patients, human and veterinary.
Mhm.
Great teams. I love working with great people. And I think my skill in science is not necessarily what I know. It's my ability to harness disparate personalities to singular missions and bring them on board and get everybody to benefit from interacting with each other. And then
That's
recognize, no margin, no mission. If you can't generate some profit, it's going to fail. If you can't support it financially, it's going to fail. So that's what drives me. So about 1993, I started doing this. I was doing it honestly. I had small children, and I was looking at ways to increase my revenue, and I started consulting in industry. I had a unique background in physiological optics, comparative anatomy, cell molecular biology, wound healing. I could bring a unique blend to a project for a company that I think is not present everywhere.
Mhm.
And when we saw this opportunity to start OSOD, I looked at it and I bootstrapped it with very little money, and I did it project by project. And I can tell you, when I started OSOD, I built my own booth. I packed it up, I shipped it, I unpacked it, I hung it up, and I sat in it at
Mhm.
the ARVO meeting. And I did that for many, many years. And when I started, I took no money out from anything. And I used the money that I generated to help support my research habit. And I took the money into the university, and I generated a fair amount of money that allowed me to not suffer the dynamics of up and down funding.
Mm.
I had a cash pot that I'd never had to fire people.
Mhm.
I could ask questions for which there was no funding for that I found was interesting. I studied refractive state of fish in nocturnal conditions and understanding how there was movement of the photoreceptors. I found that interesting. I could pay to have that done. Nobody would fund me for that. I studied vision in dogs. For 30 years, I studied vision in dogs. Why? Because I love dogs and I'm intrigued by vision. And I do have a background in optics and visual capabilities. And I never had any funding for that. But I had an ability to study that because I had discretionary funds that I generated from industry. And then when I privatized Ocular Services on Demand, it was with the blessing of the University of Wisconsin, because when I started it, I went to the university lawyers and I said, this is what I want to do. I want to make sure I do it correctly. I want to do it transparently. And at that time, the lead lawyer for the university told me, if this works, you're going to have to leave the university. I said, why? She goes, because we're not going to be able to keep up with it. And she was exactly. When we privatized OSOD, we were generating so many CDAs, so many MTAs, material transfer agreements, confidentiality disclosure agreements. We were drowning the central office. And so they congratulated me and said, it is time. That's great. Privatize. And we did that right around when I came to Davis, where we privatized it. Until then, every penny went into the university. And again, because that was in the mission as to why I became engaged. And then when I started OSOD, then it was like, I really like this. It was a whole set of skills that I had no experience with. Then all of a sudden, I'm in charge of a group. I knew how to run groups and how to focus people on missions. But dealing with the finance, the banking, the insurance, the contracting, the never-ending things you do in business was a whole new skill set. I wrote my first business plan using Business Plans for Idiots. And I bought two. There's idiots, business plans for idiots, and business plans for dummies. I sold them, and I wrote my first business plan, and I raised money. OSOD never had to raise money. I had to raise money for every other company that but I was really green. And you make a lot of mistakes when you start things. And the mistakes I made when I started out in different companies positioned me for success in everything else. Now, some of the companies didn't knock it out of the park, but they did okay. And they helped, and societal good is helping to employ people. I've been able to employ hundreds of people over my lifespan in business. And I feel there's a lot of mortgages, and we have put a lot of kids through college. And I feel really good. And OSOD is a crazy business structure that I built because it was built primarily to benefit the people who would answer the phone for me and show and do things. Over 80% of all the profits in OSOD went out to the people who were our consultants. Always had been. And the profit that we made within OSOD benefited the University of Wisconsin. And then later, a lot of the money that we generated, we directed some of the money to benefit the University of California, Davis. OSOD helped buy things for universities because they were missions, because our consultants asked for instrumentation that would help their research. We'd come up with that. If we could have a rationale where it could help them help in OSOD, but also help the academic mission, help society, we would do it. And we've never been, no entity that I've been involved in has been primarily driven by profit. Now, again, no mission, no margin, no mission. We understand. But it's not been primarily to generate money.
Yeah, look, we're big believers at Natera that you can do great in the world and make money at the same time, right? And that's always been our kind of mantra. So I noticed OSOD, you introduced it as a merit company. Are you able to kind of disclose a little bit more about what that's all about? I know there's
Sure,
a
sure.
affiliation there. Yeah.
Okay, so going back in time, OSOD has this model where we harness expertise to address issues in drug development and ophthalmology. Well, OSOD is focused on preclinical development from molecular biology right up to investigational new drug applications. We can help companies start to finish. I mean, from in silico to in vitro, cell molecular biology, we have expertise all the way along the line. Currently we have 11 veterinary ophthalmologists. We have multiple physician specialists that are part of OSOD focused on preclinical development. But we have a lot of expertise in knowing what the translational moments are. And what I always recognize is we'd get a company to IND and then we'd say, see ya, best of luck. And we had a number of members of our group that are physicians that were like, why are we not part of that, the clinical trials? And long story, very long story abbreviated, is I courted Ron Danis and Yijing Wang to start a continuity of OSOD into the clinical arena and privatize drug development and helping with human clinical trials with some unique attributes. And so Ron Danis, Yijing Wang, and I started a company called iCor at Wisconsin. Ron Danis was the director of the Fundus Photograph Reading Center at Wisconsin. Yijing Wang was in charge of imaging technology at the reading center. And Ron always entertained my discussions at this science meeting called ARVO every year, and never did anything beyond letting me buy him a beer and listening to me. And then Yijing Wang got recruited to Wisconsin, and the three of us formed a company called iCor. A very compressed history is iCor was successful. OSOD helped fund iCor. Mike Nork is my partner, and at that time it was Mike Nork, Dan Albert, Dick DeBilzik. We helped fund iCor, the formation of iCor, and I was a principal in iCor. And I helped set up the infrastructure for iCor. And then we moved along. We became profitable in a short time frame. And I exited the company. There were when I was very busy, I was a full-time professor. I was running OSOD. I was also the CEO of iCor. And things got very busy in contracts, and there's that element. And then that, as happens in many startups, a disparity of views as to how things should go. And I exited iCor. And then we still maintain good relationships. And actually, iCor and OSOD were in the same office space because I was the CEO of both, and we combined everything. And then all of a sudden, OSOD is now separate from iCor. And then we go, I forget, 7 years down the road. This past year in 2025, in January, I sold OSOD to what Merit evolved out of iCor. iCor was focused on ophthalmology only. Under Yijing Wang's leadership, with Ron Danis as a principal still, expanded iCor's mission to include oncology and respiratory in imaging. It's an imaging CRO, contract research organization, and they have now acquired the preclinical arm of OSOD. So I'm the CEO of OSOD, but it is owned by Merit. And why did I do that? I did that because I thought it best respected the people who've saddled up with me. I am very sensitive to the fact that I have a number of mortgages and families that are dependent on acting rationally and responsibly. And to be honest, I could have sold the company for more money, but it would not have best secured the legacy of OSOD and would not have best advantaged the employees and the consultants of OSOD. So at that time, Mike Nork and I were the sole owners, and I think we did the right thing, doing societal good, taking care of our teams that had saddled up with us. And yes, we made some money. So
That's awesome.
that's how OSOD is now a Merrick company. And my
Hmm.
plan is to slowly fade to black as soon as I'm not needed in
Hmm.
management. And I'm really a part-time manager now because
Sure.
OSOD is being integrated into the much larger structure. Merit is a
Hmm.
much bigger company than OSOD.
Hmm.
It is slowly being integrated. I'm there to help maintain the culture and the integrity of the relationships as we pass it over to Merit management. And Merit knows everything about OSOD because the same guys who started OSOD, we then, Yijing and Ron were always
Hmm.
aware of OSOD, and I applied the principles of OSOD to starting the infrastructure along with Yijing and Ron that formed the basis of iCore, which then became Merit. So again, we're still in the same office space. We didn't have to move.
Very cool. Very cool. Very
So
cool.
that's
So
how
the,
it happened.
so your world, it came full circle with this, and it sounds pretty awesome. Congratulations. That's a lot of work, a lot of blood, sweat, and tears went into that. Tell
Yeah,
tell us a little bit about Embed. So I think, look, you know, we've connected over multiple topics over the years, but Embed is something we're obviously at Natera very excited about, where we see it as a centerpiece to our advanced wound care strategy alongside some of our cold plasma technology. But would love to kind of hear the embed story from your perspective. We've also, we've interviewed Ankit in the past as well, and so it's great to hear his story as your former postdoc. And so we'd love to kind of hear the founder story, but also what happened there that got you excited. What made you want to go jump into Embed?
okay. So Embed, again, this is the university, the academic private sector crossover, which is, my whole career has been one leg in academia, one leg in the private sector, and in operating transparently within the university. That's key. And if academics get into the private sector, my advice is be transparent, challenge them. It doesn't matter if they've never seen it before, they've never done it before, tell them about it. And if they say you can't do it, ask them for the piece of paper that says you can't, because there usually isn't a piece of paper. It's That's the challenge in academia is mid-level management where it's a little bit like banking. They're not adventurous, right? So my engagement in Embed arose from getting a very large grant to put together a team to study wound healing from the cell molecular through translation into humans. And I'll remind you of 2008, 2009, where the whole world was in the crapper financially. And there were programs that were initiated to try and stimulate the economy. And one of them was to throw a bunch of money into startups and put money into research in the university. And money was flowing through the National Institutes of Health. And what they were looking for were established teams that could study innovative, world changing technologies. Okay, that was part of the mission.
Hmm.
And they gave very large grants. And I put together a team. I was the PI, and we got a very large grant because we had a large group of people who already studied wound healing together
Hmm.
at the University of Wisconsin, and I had connectivity, as did other members of the team across the country. And Michael Shore, I don't know if you've interviewed Michael, but
Yeah. Oh, I have
Mike,
not interviewed him, but I've met him. He's a great
yeah,
guy.
he's a great guy. He's a very well-known wound surgeon,
Hmm.
very, very enthused, and was a member of the team. Nick Abbott, who is a world-class chemical engineer, an interfacial scientist, meaning that he studies things at interfaces. And wound healing is something that happens at interfaces. So Nick was engaged in it. Chuck Saprinsky, who's a microbiologist and an immunologist, was part of the team. Ankit Agarwal, who is now was the founding CEO of Imbed and remains the chief scientific officer, was training with Nick, myself, and working with Chuck Saprinsky, and John McAnulty. At that time, John and I have been friends. We were all friends. We have a shared interest in wound healing, and John is a general veterinary surgeon, highly innovative, and an expert in wound healing and comparative medicine. He later on became the chairman of surgical sciences at the University of Wisconsin School of Veterinary Medicine. But we were the founders of Imbed. And we founded Imbed because we had this postdoc at the time, Ankit Agarwal, who, as part of his training, became a Kaufman Fellow. And I'm sure he was able to explain to you what that meant. And he was really driven and wanted to start a company. And we had a very large platform that we had explored in the context of this NIH grant. And one small piece of that platform was polyelectrolyte membranes. Now, it's important, again, being transparent. We filed the intellectual property before we got the grant because Nick and I and John McAnulty had, before we started this team, we had already envisioned where this would go. And Wisconsin's very unique in that they allow the faculty to apply for patents outside the university if you have not used $1 of federal funding to generate the idea or proof of principle. So before we did one experiment, we filed a patent, and of course the university came in and explored and you have to get permission to file outside the university, and they explored it with hammers, tongs, and flashlights, and would like us to file with them, but they were not willing to negotiate terms, and we decided no, we just paid for it and filed it. And that with Ankit, this is about 2009, 2010. Ankit, finishing his postdoc, wanted to start a company. We supported it. And we provided the in-licensing of the technology because the technology was held by an intellectual property company that we formed called Wound LLC. And Wound LLC, which owns a very large body of intellectual property on wound healing, and licensed a small piece of it to Imbed. And then Nick Abbott was engaged with the founding and guiding Ankit in the early stages. And John and Chuck and myself and Mike Shore helped support it. And then we've all played critical roles at different points going on, including Mike Shore at different points with Imbed. He was on the board. He's remained a strong clinical advocate. Chuck Saprinsky helping at the university and looking at things with its antimicrobial properties on the silver products and helping generate information on the anti-biofilm aspect of Imbed. John McAnulty with establishing proof of principle. And again, everything was done transparently with the university. And in Imbed, again, this crossover, Imbed helped benefit the university, and the university helped them benefit Imbed with contracts and cross flow of information. And I think that it embodied what is important to recognize is that Wisconsin is at the university is the Wisconsin idea. And that is the notion that the state-supported entity of the university should have a focus on benefiting the populace of Wisconsin. And that is the Wisconsin idea, that it shouldn't be an experiment done in isolation. It should be something that helps benefit society and locally in the state. And I think Imbed is a great example of how the university is adhering to the Wisconsin idea. And you can look this up, the Wisconsin idea. And I think Imbed embodies the Wisconsin idea.
That's awesome. Look, I love this. So I love the Imbed story. I thought, obviously it took a number of folks to get this thing off the ground, and the IP story is really interesting. Help us understand how you got involved in wound care. I know this kind of goes back to your interest in kind of multiple aspects of science and medicine, but your lab, which presumably was a Vision Sciences lab, was somehow supporting this wound care grant, right? So what was it? Was it purely just intellectual interest in the sector that got you involved, or was there some link?
So the connection here is that during my corneal fellowship, I had volume patients. I got advanced training in microsurgery of the anterior segment of the eye, and I did cell molecular biology training with Ted Reed on wound healing.
Awesome.
Studied wound healing in vitro.
And so wound healing of the eye, or wound healing?
I started off with wound healing in the cornea.
Very cool. Very cool.
And because wound healing is a process that has commonality across tissues, and I started getting involved in different aspects of wound healing in the cornea, we were then branched out into wound healing in skin.
Yeah.
And looking at a number of biomechanical factors, chemical factors, cytokines, growth factors, electrical fields, you name it, as we're looking at it. And I saw the opportunity to put together this large grant. It was about a $5 million grant to study wound healing in different models. And coming out of that, as well as the intellectual property that we had formed Wound LLC, we were able to support Imbed, which is really focused on a small attribute of our intellectual property, which is the polyelectrolyte membranes, and looking at it as an antimicrobial and helping to support wound healing itself without having any toxic events, and serving as an antimicrobial, preventing adverse events associated with the wound healing process, either created by a surgeon or happening after a burn or happening when you get, physical trauma.
So is it true that the cornea is like the Wolverine of the body when it comes to healing? Does it? I mean, I've heard that. I've heard this from folks who've gotten LASIK procedures done, right? The eye heals pretty fast.
I've never heard that. I think it's interesting. It's
Wolverine
of the body. Okay.
Yeah, maybe not the best analogy here,
I like that. It's a little bit like the werewolf story, that if you change into a werewolf and come back, your wounds are healed. Well, corneas have a very good ability to heal. It's true. As do other mucosal membranes.
Okay.
The inside of your mouth.
Yeah.
Right?
Yeah.
Your rectum, vagina, your respiratory tract, mucosal surfaces have a strong ability to heal. Part of it's related to blood supply. Part of it's related because, well, hell, when you're chewing on stuff, you're wounding yourself all the time. When you're out in the environment, your skin has a great ability to heal. Why? Because you're constantly running into things. Go back to evolution, you're constantly running into stuff. Things are trying to eat you. If you got a bite and you died, the whole species would be gone. All right. You have to be able to maintain the integrity of your ectoderm, of your barrier, because your skin, your epithelial covering on the outside, and the epithelium cover the entire outside of your body and the entire inside of your body. Your oral your oral membranes, all the way to your esophagus, along your GI tract, right to the anus, your respiratory tract. Everything is covered with an epithelial surface. And so it has to heal. It has to heal because the epithelial cells are your major barrier to being populated by microorganisms that ultimately could kill you.
Interesting. So are the secrets of healing? I mean, is there, I guess, I hear that the eye is sort of the biomarker of the rest of the body to some extent. Is there something to be, I guess you can learn it from all epithelial tissue, right? Or mucosal tissue, sorry, but
yeah, no, it's different. One of the advantages of the eye is that you can see both epithelial and stromal wound healing in real time because it's transparent tissue. You can take a microscope, you can see it, you can look at it, you can probe it. But skin wound healings, you can see it, you can see the wound healing, and just the cornea, normal humans, normal animals across all vertebrates heal really well. Why? Because they're alive. As a species, they have made it. And if you are a wuss and you do not heal well, you don't survive. Right? And so normal wound healing is actually a very hard thing to beat. So where the issue with wound healing is, and the challenges are when you have large areas of wound healing or where you have compromised wound healing because you have denervation. Turns out your nerves are really important to wound healing. They help direct things in terms of how wound healing happens, but they also elaborate. People don't think of it in terms of, oh, I got nerves. I either feel it or I don't. The nerve endings themselves provide neurotrophic factors that actually accelerate wound healing. Things like vasoactive intestinal peptide, substance P, calcitonin gene-related peptide. These are all things that are elaborated at the nerve endings that accelerate and direct migration, proliferation, and adhesion, which are the three elements that are most needed to successfully heal a wound.
Awesome. Gosh, I again, every single time I have a conversation with you, I learn something new. So absolutely fascinating. And thanks for giving us very comprehensive background about what got you into to the industry you're in and where you're at now. I'm going to kind of change things up a little bit with our interview here. I'm going to get into some more of sort of like your personal take on things, right? Not necessarily your personal life, but like your personal take on lessons learned, challenges you've faced over the years, right? So let's start with kind of leadership and values, right? I mean, this is a big part of who you are, and you are one of the most ethical business people I've had the pleasure of working with, and I'm not afraid to say that publicly, because you really are a great person. But how does your experience in academia inform the way you lead some of these companies that you're a part of?
Well, science leads. Science leads. And, I've sat through meetings with companies, with leaders in companies that the plan leads, their hopes lead, or investors lead, and science leads,
What?
By this, here's what I mean by this. Every time you go off and you start with an asset, you have a vision. For me, it has to be motivated by science that's innovative and strong, right? And then you build great teams around it. And then you have to have market pull. That's something I had to learn earlier on. You can't just build something without understanding that the market really wants it. If you build it, they will become that. Well, that's great if they need water and you're offering water. But, if you're building ice skating rinks in Alaska, that may not be what you need, the populace needs. So science leads is one thing. And you got to trust the science. If it's well done, you have to know when to hang it up, that the project isn't working And that's maybe a very hard thing. And I've seen companies make this mistake many times that,
Mhm.
oh, we failed at this, and it's usually a clinical trial or, the IND enabling trial that's done in a GLP environment, good laboratory practice environment, very disciplined, uh,
Mhm.
and it didn't work. Well, then, if you have a company that then goes back and you do have to go back and ask, is there another avenue? But if it's not a clear avenue and it's a desperate take, sometimes you just got to know when to hang up. It didn't work. And you can then
Interesting.
look at what the question you asked and answered said no, then you have to hang that up. And then you can look at the sidebars off of that and
Mhm.
say, well, maybe it didn't work for this, but will it work for that? You can do that. And that's if you're an investor and I gave you $1 million, I expect you to do that
Mhm.
because I
Mhm.
still want you to try and do everything you can to realize a profit for me. But at the
Mhm.
same time, you can spend more of my money by not saying it failed, you know,
Mhm. Mhm.
desperation,
Mhm.
so
Mhm.
I think so. For my principles, as I already said, the hat trick is societal good, great teams, and margin and mission. Great teams
Very
is
cool.
critical in that. And if I look at it, I guess I can disclose here, I'm also an active investor and yes, I'm a manager, but I'm also an investor in different in companies at different stages. And I look at what the market opportunity is. I look at the asset itself, and I bring in teams of scientists to look at the asset and kick around the data room. And I bring in scientists I trust because they're solid and they can look at data and they can tell whether or not it's optimistically interpreted versus cold hard facts. I like cold hard facts. And I invest as much on the team as I do on the asset and the opportunity. And if I don't see a team that is cohesive and has the experience to enter into the field and understands the elements, I won't invest. I don't care how good the idea is. I don't care how big the market is, because the team ultimately is who's responsible for bringing it there, bringing it across the finish line. So
That's awesome.
I put a lot of faith in my ability to analyze the integrity and the capabilities of a team. I spend as much time assessing that as I do kicking around a data room in order to determine the strength of the proposal. That's what the data room tells me,
Mhm.
the strength
Mhm.
of the
Mhm.
proposal.
Mhm. Mhm.
But then
How
I
how do
spend,
you how do you evaluate a management team though? How do you what's your philosophy there?
I try to meet with them personally. I
Mhm.
reach out to people around them.
Mhm.
I have a large network. My teams I'm associated with have a very large network, and I ask everybody.
Mhm.
And if I go into a data room, I will bring in 3 or 4 people from my group, either from OSOD or scientists I trust in that area. And
Mhm.
if I'm not an expert in the area, somewhere in our group, we have somebody who knows somebody. I'm from New York, right? I know a guy. And if you're from New Jersey, I know a gal who knows a guy. And, so we exercise that. But assessing the quality of the team, it's really about, the history is the best predictor of the future. Do we have a seasoned individual, somebody who's a leader who has found success? And if they didn't meet success, did they meet success with high integrity or fail with high integrity? That there's not a wake of complaints in their exit, in the closing of their effort. That when you talk to people, I was in vet, I'm not going to name names, but they're friends of mine. I invested, didn't work out. They had a great plan, great, great
Mhm.
invest invested. They ran a quality trial. I looked at the science behind it. I understood it. And it was
Mhm.
done in a very short period of time and it did not work. And we put it in the loss column, all start to finish,
Mhm.
out of a human clinical trial. Who does that? Nobody.
Right.
because these guys were seasoned, and so failing with integrity is part of the history. If they failed, I want to hear that they failed with integrity and with real diligence on it, that they failed for the right reasons. And if they're
Gotcha.
successful, I want to hear what the people adjacent to them say. I want to know what other investors think of them. I want to know what, if you're talking to people in VCs, what the other VCs think of these people.
Mhm.
If it's a VC, I want to know what other startup leaders think of the VC. You know,
Sure.
it's an imperfect art and you can fail. And I have failed in assessing people, but not very often.
Yeah, I think we all have. Yeah, we all have. It's tricky. People, you know, I think you've said this to me before. Businesses are easy, people are hard. So people can be tricky to read at times, and it is a very it's a difficult thing to do that we're always trying to figure out how to scale. But
There are
well, let
and
me
there are people who can present themselves extremely well,
yes.
extremely polished, and in the back seat they ain't got the goods.
Yeah.
That's
That's the
hard to
trick.
sort out. That's
That
where
is that's
the
right.
people looking at history. I always like seeing somebody, being an academic. I can tell you, academics, we're great at ideas. Oh my God, are we good at ideas? Well, ideas are cheap. You can't, I got lots of ideas. Everybody's got lots of ideas in academia. There are some great ideas.
Yeah.
Ideas are cheap, development's hard, and commercialization's impossible. And to
Sure.
invest, I want to see that somebody has been the road of development to commercialization. If you're missing links, maybe not holding. I may be holding on to my money.
That's very helpful. So what advice do you give academic scientists who, they want to become entrepreneurs, but they feel overwhelmed by the business side of things, right? What advice do you give those academic scientists?
Pay attention to the team you build.
Hmm.
Don't.
Hmm.
I made the mistake. Fortunately, OSOD was successful,
Hmm.
remained successful, but
Hmm.
oh my God, we had no idea what we were doing in business. And
Hmm.
I learned through bumps and mistakes, but I always had a clear vision in mind. And I am tenacious, and I do learn from my mistakes. I'm trainable. I like to think I'm still trainable. My advice to academics would be the following. One is, are you sure you want to do this?
Hmm.
Because a startup is an all-in exercise. It is not, and if it's a hobby, don't do it. Don't do it because it's not going to work. If it's a hobby and you don't have somebody who's, I always give the image of every mission, every startup has to have a heart that beats that drives it.
Hmm.
And it ain't a part-time heart. It has to
Hmm.
be, it's an all-in exercise. And, I could speak as someone who's done all this while raising two beautiful daughters that are both
Hmm.
successful in their own right, being married to a woman who's every bit as successful as I've been in any context. And I think if you ask the right certain people, they'd say she far surpasses me in many aspects of what she's done. She's double boarded. She's a founder of a college in the profession of veterinary medicine, all this stuff. And my experience of startups is that you have to have the ability, if you're going to stay full-time and stay successful in academia and all the pressures of academia and do a startup, you have to buy into the fact that you are going to be working 70 to 100-hour weeks.
Yeah.
And if
Yeah.
you're not willing to say you're going to do that, I'd say don't do it. If you're not willing to experience the challenges and the pain of having your time sucked out of you, really think hard about it. That's one. Two is don't leave the university too soon.
Okay.
We made this mistake with the company. We had great ideas and we still have great ideas. And naive enough not to see how long it would take to commercialize and not understanding. Remember, ideas are cheap, development's hard, commercialization's impossible. Oh my God. And Imbed taught me a fair amount. Oh my God, the commercialization piece of this thing was in healthcare. There are so many levers that you have to pay attention to and forces you have to pay attention to that I had not fully considered. And, again, I was a founder in Imbed, and then I wasn't part of the management that developed Imbed. I was an advisor, call me for an opinion, but I was not on the day-to-day management and on the floor administration. And it was a good thing because I didn't have a clue about how do you get into hospital billing. We required a whole different skill set. So I think leaving the university too soon is a little bit, the analogy would be that you're throwing the ball too far down the field ahead of the receiver, not ready for the reception. So stay in the university and take advantage of the people around you to understand when it's time to leave the university with your idea. Understand that if you want to exercise the federal funding route in SBIRs, which is a terrific road, STTRs and SBIRs, terrific road to go, understand you need somebody who's at least 51% with the company. That's got to be the heart that drives it. And if the individual, whether, is a scientist or a clinician or, anybody in academia, if you're not willing to step out 51% of the time, and I understand why you wouldn't. I never stepped out from the university the whole time. If you're not willing to step out, you need to find somebody who's willing to drive it. And don't step out until you have identified that person and recognize that if it's just one person, that your enterprise is at risk because it's dependent on one person. So the third thing I would tell you is pay attention to integrating redundancy in your plan, in that you want to have it that you're not dependent on one individual. With your plan, that be actualized. And the last thing is you need to understand market pull. Where is the market pull? Where can you go and get reception inside of 3 to 5 years? Now, when you start off, you're thinking, oh, this is going to be a cakewalk. I'm going to be going here in 2 years. And I'll tell you, my experience has been 7 to 13 years, 7 to 15 years. Nothing in under 7 years, unless it's already pretty baked idea, and you're not going to commercialize it. You're gonna exit it to a strategic, a company that wants to buy it, that they're going to commercialize it. So commercialization is a whole nother thing. So have a very full plan put together that has reality checks around it before you start.
That's awesome. Gosh, that's comprehensive advice. I'm sure whoever's listening with an academic background, I get this question all the time, right? From professors and entrepreneurs with academic backgrounds, and they that's the hardest thing to figure out is get a plan in place and surround yourself with really smart people that can advise you and guide you along the way. But I think that was fantastic. I want to change things up a little bit. I want to dive into some kind of challenges and reflections over your career. So kind of first question here is, what's been one of the toughest decisions you've ever had to make in your career, either as a professor, scientist, or a CEO? You choose.
A decision affecting me or affecting other people? What are you talking about?
So professionally. So what's one of the toughest, I guess, business decisions that you've made in your career?
Business decisions. Selling selling a company, selling iCore.
Mm-hmm.
Well,
Mm-hmm.
not exiting. For me,
Exiting.
exiting iCore, that was a very hard and emotional decision. Selling OSOD, but that was easier because I sold it when I was 69.
Mm-hmm. Mm-hmm.
I'm
Mm-hmm.
70. I'm
Mm-hmm.
70 now.
What goes into that decision though? So is it the emotional pull? You have a personal tie to the company, it's your baby and you don't want to sell it to someone, or is there
Well,
something else that goes into
every
it?
transition has got different motivations,
Mm-hmm.
or my motivations for leaving iCore are very different than selling OSOD to Merit. You
Uh-huh.
know, I could, and then when you start anything, these are big decisions.
Uh-huh.
Signing up for
Uh-huh.
things are big
Uh-huh.
decisions.
Uh-huh.
I mean, having a lab of 30 people and teaching a bunch of residents and postdocs and doing, that's a full-time job. And
Right.
everything you pick on is like every one of those decisions is a big decision because it's zero-sum game in terms of I like to think that there are only so many experiments I can do in life. And so I really question every experiment I do and say, is this an important experiment? I wake up every day and I challenge my trainees and the people that work with me in the private sector is have you identified the most important thing that you're going to do today? And if not, that's the first thing you got to do is identify the
Mm-hmm.
most important thing to do
Uh-huh.
today. And then
Uh-huh.
you don't do anything until that is done. And everything else is secondary. You don't do it. You do the first most important thing first.
Yeah.
And and I've had lots of academics and that I've worked with over the years, and they want to explore this or explore that, and they come to me with an idea that they can get done quickly.
Yeah.
Like, okay, but why would you do that? Well, because I can get it done quickly. It's like, well, why are you doing it? You need a publication, or is and if you need a publication, I get it. That's part of the uh, the coin of the realm in academia, publications, grants, all that. I get it. If you need it, I get it. But you have to ask yourself is that the most important experiment you can come up with that meets this need? Is
Yeah.
that the most important question to ask? And it's really bringing that discipline early on. I wish I had been given that advice when I was in my 20s. Focus on the most important thing first. Is it the most important experiment? Is it the most important paper to write up? I would write things up, at that time this is back in the 70s and
Yeah.
the 80s. It's like,
Mm-hmm.
I'd write it up because I could. Hell. And everybody, at that time, people were pretty impressed that you could write a paper and get it published. And I would have been well served by having that discipline that I have now, really saying, is it an important experiment? Does it change things? Is it important to the field? Is it important to me? And is it an end of it in and of itself? If it's an experiment that's a one and done, I'm very less interested in it. Unless that experiment is so important that one and done informs the field and changes how people go down that road. So I
Mm-hmm.
always ask, what's the longevity? Why am I doing this? And the second question has to be, what does it lead to? It's same with companies. If I'm starting it, what does it lead to? What is my vision? What is my 3 and 5 year vision? Recognizing that my 5 year vision is probably totally wrong because everything changes. But where is the mission going? Not just
Mm-hmm.
the task, but the mission. I try to take any task and integrate it into a larger view. And for myself, I use the word mission. What is the mission? Here's a task in the mission. Why is that task important? And how does it help me achieve this mission? And if it doesn't help me achieve the mission, I won't do it, even though it's an interesting thing.
That's awesome. That's awesome. That's good perspective to have. In terms of legacy that you're building, right? Because you are a legacy man for sure, right? Everything you've ever done, you're very intentional about how you approach people and the things that you build. And you've mentored countless students, clinicians, founders over the year. You mentored me. What's the legacy you hope to leave behind, right? Not just through companies, but through the people you've interacted with.
Yeah. So in academia, and I've been retired now from academia in 3 years. I still get I've become, to use the Godfather terminology, I have become a consigliere. I get reached out to for managing challenging political terrain in academia.
Mhm.
Challenges, occasionally I get calls specifically on an experimental design or something, but it's really about career. How do I manage this? How do I navigate this? And I would say when I look back on my career, the thing I am most proud of in academia are the people I've trained and the teams I built and the culture I created. And I believe that the culture I created is nourishing and has helped the individuals build their careers, mentor their students in ways that benefit the students. I take a very Confucian view of leadership in that I believe that leadership is best accomplished from behind. And when you're willing to give people credit and not take it yourself, it is unbelievable what you can accomplish. So that culture, I've had many, many trainees, and I like to think that that is moving forward and paying it forward down the road. But did I change medicine in academia? Honestly, maybe not so much. I moved a lot of things forward a little bit
Mhm.
because of my academic intention. It led to private sector activities. But in the private sector, we helped change the practice of ophthalmology. We provided support. And again, I'm going to mention a few products out there because we published on them. But we provided the infrastructure, scientific support that allowed for a successful IND for Lucentis, for Eylea, which are the two major treatments in the world today for age-related macular degeneration in humans.
Mhm.
And it changed retina practice, and I'm a doggy eye doctor. And that activity changed medicine to a degree. We helped provide the infrastructure and the scientific support, leading to a successful IND that led to Xiidra, a anti-inflammatory that is used for the treatment of dry eye. Chuck O'Neill, I'm going to mention Chuck O'Neill, and he's been important in my journey because Chuck was the lead toxicologist for Lucentis at Genentech. He then became the vice president of BioMarin, which is focused on predominantly rare diseases, vice president in charge of preclinical development. And he allowed me to recruit him. He was well aware of OSOD because he interacted with it so much. But he's on the executive team for OSOD. And he's like one of the, and we talked about team. He's an individual that I point to all the time. This guy knows translation. His whole career has been preclinical activities that are predictive of being successful in human clinical trials. And he has, he walks the walk after he talks the talk. He knows what he's doing. And he's an individual that I work with on a regular basis. Again, a team. Craig Crossan also on the executive team. Gary Leatherberry on the executive team. Craig Crossan, a very successful academic, a premier ocular pharmacologist and physiologist. So building, it comes back to those teams in academia that then I translate over to the private sector. In the private sector, I'm very proud of the teams that we built, the kids we put through college, the mortgages we supported, and the investors who we made feel smart for investing in us. And I'm hoping Imbed is going to be one of the great success stories.
It will be without a doubt. It will be without a doubt, and we're very excited about our investment in the company and our continued support for the business. So Chris, we've got we've had a lot of great conversation here. You've spent your career at the intersection of science, vision, and innovation. You've done so many extraordinary things with deep humility and impact. As we wrap up, I'd love to ask you two final kind of more personal questions. Number one, was there a single idea, book, or mentor that's been a steady guide through all these phases of your journey?
Yes. Yeah.
Tell us more. Tell us more.
So I would tell you that I have been strongly influenced by the teachings of Confucius.
Confucius. Okay, cool.
Tell
us more.
There's an interpretation of, Confucianism can simplistically be in Oriental philosophy in general. I studied in undergraduate and it influenced me. But I studied Confucianism and the analogs of Confucius. There are parts of Confucianism that inform how I do science and how I train people. There's a big focus on ritualism, the execution of ritual. And, there's one way of paraphrasing one of the analects would be that a ritual performed without high skill is clumsy, and without presence is empty. So I believe being present in the moment is essential as a clinician and is essential in business dealings. That it's not just, I'll give you an example. You walk in, you're going into an important thing, and somebody says, "Hey," and they look away. Versus you walk in, somebody looks at you, recognizes you. You can feel that they're present, and they say, "Welcome." That's what I'm talking about. Another analogy to this would be, you've seen the same performer perform twice, whether they're a singer or an actor. And one time it felt like they dialed it in, and the other time it was transformative. What's the difference? Being present. And being present, and whether it's in clinics, and I'll give you another example from one of my mentors. You talk about mentors that affected my life. Mark Manus affected my life. Ted Reed affected my life. Howie Howland at Cornell affected my life. Walter Dewey, as a investor and as a financial manager, and affected my life. And all of them, I would say, were present and with high integrity. So Confucianism, to me, provides a patchwork of guiding principles that I integrate into how I train people and how I do surgery and how I treat people in business.
Amazing. That's awesome. That's great, great advice. And, as I reflect on my own career, some of the best leaders that I've met, yourself included, are present, right? You feel their presence, and you could tell it being present, creates this aura of confidence, right? So if I'm being led by someone, I have more confidence in someone who's present, and I also feel more engaged because I feel like they care, right? So the empathy factor is really, really important. I think that's part of human nature. So I like that. I'm going to have to check out Confucianism. What is it called? Confucianism? Confucianism. Yeah.
Confucius, Lao Tse with Taoism. But if you look at the little book that I like in particular is a small book written in the 70s, written by Fingerette. It's a small
Fingerette. Okay.
Most of the residents that I've trained have had to read sections of Confucian, of Fingerette's interpretation of Confucius.
Confucius.
Because you can get bogged down in Confucianism. I would point people to Fingeret's interpretation. And the other thing that I would say influenced me is my family, how I was brought up. I was brought up by a very unusual, empathetic woman who recognized with 7 children that we were all different and expected the best of each of us, but the expectation differed for each of us. And she expected the best out of me in one way and out of one of my brothers and sisters in a different way, because she recognized that everybody was different. And I try to manage individuals knowing that. And people come from different cultural backgrounds, different religions, different ways that they practice religion, how their family treated. Every family has broken elements. Some are really challenging, and recognizing that the person in front of you really different take on the world than maybe you do. And how do you harness them to a mission to benefit them and the mission itself? It's that intersection, because I'm not the right person to work. And when I interview people, I tell them I may not be the right person to work with. Can I help you and can you help me? It's really finding that commonality. And if we can't find it, you have to recognize that if you have certain expectations that I'm not going to meet, and I'll tell you what they are. What I'm going to expect the very best out of you every day. When you come in the morning, you're going to be smart than you went home at night if you're my resident. And if that's not okay with you, you maybe shouldn't train with me, because I'm going to hold you to that every day. I hold myself to that. So I think understanding that, when you think people influence me, it's my family, that really the family that raised me has set the foundation of who I am. And everything goes from that. And then the people I mentioned, Howie Howland, influenced me along 10 years at Cornell, very influential. He treated me as a colleague, not just as a student or a minion. He treated me as an individual, brought me into his family. Ted Reed, same thing at Davis, taught me the wonders of cell and molecular biology. I'm actually hosting him and his wife in a few weeks up in Oregon. Ted was a full-time professor at Texas Tech until he was in his 80s, and he was the head of the biomedical engineering interface into his 80s. Mark Manis, I would say, changed my life because he saw 50 patients a day when I was his fellow. And how do you do that? And his patient would take a bullet for him. And by watching him, it's because he gave them a moment in each visit where he was fully present. You could feel it in the room, that nothing was more important and nobody was more important than that patient in front of him. And I learned from that.
That's awesome. That is amazing. Dr. Murphy, I just want to say on a personal note, your work has not only advanced science, but changed how people and animals see, live, and heal. It's incredibly rare to meet someone whose legacy spans both medicine and meaning. Thank you for sharing your time, your insights, and your heart with us today. It's been an absolute honor having you on the podcast. Thanks for joining us.
Thanks for having me. You're way too complimentary.
It's that humility.
We all struggle. We all have challenges. And I appreciate a chance, if there's something I'm well known for, is I'm willing to give guidance and advice at no cost, and the advice is worth every penny they pay for.
Thanks for being on. Appreciate your time. Thank you for joining us on the Health Tech Invest podcast, powered by Natera Ventures, your go-to source for healthcare and tech venture investing. For additional information, resources, and ways to connect with us, please visit nateraventures.com. Together, let's spark innovation for the future of tech and healthcare investing.